Shalom and welcome. This episode will cover the question, what is Abraham and the ancient Israelites' Y-DNA haplogroup? We will specifically speak from the perspective of the paternal descendants of Abraham, because I'm aware that other lineages and haplogroups could have been among the Israelites, but for this video, our main goal is to look for the direct bloodline lineage of Abraham and his descendants. What will be their haplogroup? With that being said, what is a Y-DNA haplogroup?
As many Americans know, the third president of the United States and author of the Declaration of Independence, Thomas Jefferson, was an important figure in the emergence of early American identity and is considered one of the major founding fathers of the country. And although many today argue over his own personal moral character and ethics, it was a major point of contention even hundreds of years ago, and many spread rumors of Jefferson's ongoing affair with his slave Sally Hemings and whether he was the father of her 6 children, an act which would have been highly frowned upon even in the 18th century. Centuries later, in 1998, it has seemingly been proven through DNA tests that Jefferson was indeed the biological father of Hemings' children, despite the fact that there were no viable biological samples of the deceased president in existence. So how did they do it? When it comes to human genetics, perhaps the best way to determine hypodescent or common ancestry in individuals and population clusters is through a specific genetic marker known as a haplogroup. Haplogroups are a common way for geneticists, archaeologists, and historians to make conclusions over the origins, exchange, and evolution of people groups all over the world, and you may have heard me mention them in videos from time to time for the past year, but unless you knew beforehand, information on haplogroups may just cause confusion and misinformation for many viewers. Therefore, I've created this video to better give an understanding of how haplogroups relate to genetics, race, ethnicity, community, and other aspects of humanity that paint such a beautiful picture of the history and diversity of our planet. For as long as can be traced back, all females carry a maternal haplogroup inherited from their mother, while males carry both a maternal and paternal haplogroup, seeing how men have an X and Y chromosome. The paternal haplogroup inherited by the father is known as the Y haplogroup from the Y chromosome, while the maternal haplogroup is inherited from mitochondrial DNA and is thus referred to as the mtDNA. Because of historical patterns of migration and the nature of male-female relationships for most of human history, generally speaking, paternal haplogroups are better studied, understood, and mapped out, although this doesn't make maternal lineage irrelevant or any less interesting. For this reason, haplogroups will only survive to the next generation if they're passed on to their offspring of the same sex through the X or Y chromosomes. Now, you may not necessarily ever have an offspring that is the same gender as yourself, in which case your specific haplogroup would die with either you or your child, but at least one of your parents sure did, as with one of their parents and grandparents and great-grandparents and so on, back to the genesis of the human species. Barring genetic mutations, if you're a male, it's an absolute certainty that you will share the same paternal haplogroup with your father, all of your biological brothers born of the same father, all of your paternal uncles, any paternal male cousins born to your uncle, and of course your paternal grandfather, his father, and so on, and all biological male children of yours will share your paternal haplogroup. Similarly, barring genetic mutation, regardless of gender, you will share the same maternal haplogroup with your mother, all of your biological siblings born to the same mother, all of your maternal aunts and uncles, any maternal cousins born to your aunt, as well as your maternal grandmother, her mother, and so on. And if you're a female, then all biological children of yours will share your maternal haplogroup, although only your female children will pass it on to their offspring. Now, you may be wondering, how did the first haplogroups form? What is the criteria for categorizing various haplogroups? How and why did the haplogroups we see today become scattered across the planet? All good questions, and as usual, the simplest explanation is often the correct one, as indeed all living humans today share ancestry with a common ancestor in both the male and female line, dubbed the genetic Adam and Eve of the human species, and the descendants of these two evolved into the 7.6 billion people of Earth we see today, and the genetic adaptations and natural selection form the modern races of humanity. New haplogroups are created when slight mutations occur when passing DNA to the next generation, and these mutations can generally be traced back to the original source, and hence the standard naming convention for haplogroups is a letter of the English alphabet, followed by the ordering of whichever specific group or subclade its mutation belongs to. In conclusion, haplogroups, along with language, ethnicity, and religion, are one of the best ways of accurately defining population groupings of the human species, and I hope this better explained haplogroups, DNA, and human genetics, and showed you just how interconnected the world's population is, even if every group has its own unique traits, history, and culture. Please let me know.
What's up, everyone? I'm Helena Alamu, and welcome to another episode of the DNA Download, where we explain the science of genetics so you can live in the know. When people talk about their DNA results, I keep hearing the word haplogroups. Joining me today is Dr. Samantha Esselman, a scientist from the 23andMe Ancestry team who can unpack this concept. So, Samantha, what are haplogroups? Truly, it sounds like a word my dad would make up in Scrabble.
Well, it's definitely a real word. A haplogroup is a grouping or an assignment of similar sets of patterns in people's DNA. So it's kind of like if we were to group people based on something like their zip codes.
Oh.
And then we can use these patterns of DNA to trace the ancient origins of your ancestry.
Okay, so then how do you make these groupings from DNA?
So people have two special pieces of DNA which we use to define two different kinds of haplogroups. The first kind is paternal haplogroups traced through Y-chromosomes, which get passed exclusively from fathers to sons. And the second kind is maternal haplogroups traced through mitochondrial DNA, which gets passed from mothers to both sons and daughters.
Okay, then what makes these two pieces of DNA special?
So that's a great question. It's because the inheritance pattern is really simple, especially compared to the rest of your genome. And actually the beginning of the word haplogroup, haplo, comes from the Greek word aplos, which means simple.
Oh, well, that's a good memory jogger.
Yeah. And so most of our DNA comes from a sort of random shuffling of our parents' DNA, which can make it difficult to figure out which side of the family each piece comes from.
Oh.
But then Y chromosomes and mitochondrial DNA don't get shuffled.
But then if they aren't shuffled, why aren't everyone's haplogroups exactly the same?
That's a great question. So over the more than 200,000 or so years of human evolution, the occasional DNA changes occurred and got passed on, creating new haplogroups.
Okay.
I have an analogy.
Okay.
So imagine a guy thousands of years ago, and this guy has a special recipe for his, award-winning paleo soup.
Oh, okay.
And then he, along with his brothers, inherited this recipe from his dad, who got it from his dad, and all the men in the family faithfully stick to this recipe, no changes.
Ooh, well, I could go for some paleo soup right about now, but please, go on.
Yeah, this analogy always makes me hungry.
Okay, good.
And so then many generations later, a great-great-great-grandson adds some cilantro.
Oh.
And then this great-great-grandson passes on a slightly modified version of this recipe to his sons. And all the men on his branch of the family tree start following this new cilantro recipe.
Oh.
And so now if you think about it, there are essentially two soup haplogroups. And this is pretty much how haplogroup inheritance works.
That makes a lot of sense. And since I'm a woman and I don't have a Y chromosome, how can I trace my paternal haplogroup?
Yeah, so what you can do is you can learn from relatives who share your paternal line. And so this would be something like a dad or a brother.
Okay, cool. So then what about the mother's side, the maternal haplogroups?
Right, you can trace this other kind of haplogroup through the DNA found in our cells' mitochondria, which turn the food we eat into usable energy for our cells.
Ah, the powerhouse of the cell.
You know your biology memes. Yeah, so this mitochondrial DNA gets passed from moms to both sons and daughters.
Well, thanks, Mom. So how can we use haplogroups to trace our ancestry?
So closely related haplogroups tend to share geographic roots, and so scientists can use the kind of modern distributions of haplogroups around the world to trace some of these major migrations. And then these can help you understand where you fit into this ancient family tree of all humans.
Wow, I feel like the world just got a whole lot smaller. Thanks for being here, Samantha, and see you next time on the DNA Download.
When you get your DNA tested for genealogy, you will often see the term haplogroup come up. What is that? Join me on today's Ancestral Findings podcast, and I'll talk on what is a haplogroup and how does it pertain to your family tree. If you get your DNA tested, chances are you will come across the term haplogroup. What is a haplogroup, and how does it pertain to your family history? At its essence, a haplogroup is an ancestral clan. Some clans are the Vikings, Native Americans, all tribes, Celts, Aboriginal Australians, and other such groups. Your haplogroup tells you where your ancestors came from deep back in time. There are also male and female haplogroups, so you can see where your male and female sides of the family originated back in prehistoric times. As with Y-DNA, which traces the male line from father to son, and mtDNA, which traces the female line from mother to daughter, haplogroups also follow straight male and female descendancy lines. Your haplogroup's point of origin is usually shown on maps at the various DNA companies that do testing. The haplogroups that exist today originated with people whose descendants were very successful at reproducing in large families. Haplogroups today are divided into four main ones: European, African, Native American, and Asian. Within these haplogroups are many sub-haplogroups that further define where a person's earliest known non-African ancestor on the male and female sides of their family originated, and when. Most haplogroup subgroups are plentiful. The only group with less than 3 subgroups is the Native American male group, which only has 2 subgroups. Basically, haplogroups chart the clans from which the modern human race originated. Haplogroups are named alphabetically in order of discovery. The subgroups of haplogroups are named with letter and number combinations, indicating where and when in time they were discovered, which main haplogroup they descended from, and sometimes even the very specific small geographical area where their earliest members lived. The more letters and numbers in your haplogroup's subgroup, the more likely you are to be able to pinpoint an exact region, and maybe even a county or town. Some haplogroup subgroups can even be traced back to named individuals if the mutation that created them originated in the more recent past. For example, the infamous Celt, Niall, of the 9 hostages, is known to be the originator of a subgroup of a haplogroup within the past 1,500 years. And some people can use their subgroup to prove their descent from him. Right now, haplogroups are the only way to trace your family tree back to the time before surnames were invented. You can take your last known ancestor in a particular direct male or direct female line and connect them to a larger clan in the more distant past, knowing that their ancestors, the ones you can't identify and trace, wound back through time to the time and place where the known ancestor's haplogroup and subgroup originated. For example, there is a known haplogroup of people who originated in France about 12,000 years ago, and who are widely believed to be the people who made the famous cave paintings in the French town of Chauvet. You can look at a maternal or paternal haplogroup and subgroup and see if it matches with the haplogroup and subgroup of the cave painters. If it does, you know you are descended from this illustrious and famous group of people. If you're interested at all in where your family originated deep in time, before the age of records, then haplogroups should be of great fascination for you. You can use them to get deep into history with your family tree. This is something that was not possible before the age of DNA testing.
Lineage important in the modern day? Some say no, but biology says yes. In almost every single culture around the world, much emphasis was placed on lineage, which is referring to an individual's line of ancestors, and still is important to a certain extent today. Some people around the world are quite proud of their lineage, and others not so much, while others say it shouldn't matter at all. Various cultures have different naming customs passed down from generation to generation in order to preserve their lineage, and this may vary by country. For instance, in the Anglo world, traditionally the father would pass on his surname to his children, but in the Spanish-speaking world, both the mother and father pass on their surnames to their children. Some cultures may traditionally only have one given name, while others may have several, but the surname, or family name, is what is generally passed on to cement the lineage of a certain family. The Kennedy, Clinton, Bush, Roosevelt, and Adams families are all examples of political families in the U.S. that have arguably been successful due to the power of their lineages. There is certainly power in a name. Nature thinks so too. That's why each and every human on the planet has a genetic surname. They're called haplogroups. Haplogroups are genetic markers that are extremely important in the study of history and anthropology because they can show an individual's or collective's simplified genetic past that gives clues to how different populations interacted. I've explained this in a past video, but essentially a woman will pass down her mitochondrial or maternal haplogroup onto all of her children, while a man will only pass down his Y-DNA or paternal haplogroup only to his sons, because only males have a Y-chromosome. Your maternal haplogroup is passed down from your biological mother to you, and it was passed down to her by her mother, your maternal grandmother, and your grandma's mom, and so on and so on. Since only males have a Y-chromosome, if you were male, your father passed his paternal haplogroup to you, passed down by his father and his father before him. Are you starting to see how this mirrors many of the naming customs from around the world? Geneticists who are far more knowledgeable than myself on the subject have been able to map out haplogroup lineages around the world going back tens of thousands of years, which gives massive insight into past human migrations, periods of intermixing, and conquest. For tens of thousands of years, these haplogroups have been spreading across the globe, splintering and diversifying due to genetic mutation between generations, and this paints a very interesting picture. This is a pretty awesome map showing paternal haplogroup distribution around the world, but
in simple terms, if you still don't understand what a Y-DNA haplogroup is, a Y-chromosome is passed down from father to son. According to livingdna.com titled, "What are haplogroups?" it reads, "The paternal haplogroup relates to your Y-chromosome, and since that is a sex-determining chromosome for men, it is passed down from father to son. This is the perfect tool to use when trying to determine paternal lineages. Therefore, if we're looking for the Y-DNA of Abraham and his descendants, we have to use haplogroups. Abraham's Y-chromosome would have been passed down to his son Isaac, to his grandson Jacob, whose name was changed to Israel, and then the Israelites themselves, who paternally descend from Abraham and will carry his Y-DNA haplogroup. With that being said, let's look at the first and one of the best candidates, in my opinion, for Abraham and the Israelites' Y-DNA haplogroup. Abraham and the Israelites could have carried E-P2, also known as E1B1. One method you can use to determine the haplogroup of Abraham is by looking at subclades of the ancestors of Semites, like the Natufians, and look at people who claim descent from the 12 tribes of Israel. One of the best candidates for Abraham and the Israelites is Y-DNA haplogroup E-P2 E1B1, which was found among the Natufian remains. In the supplementary information of the genetic paper titled "The Genetic Structure of the World's First Farmers," we can find a sample titled E1B1 among the Natufian remains in Israel. It reads, "This individual was derived for mutation P179, defining haplogroup E1B1, and upstream mutation M5403, defining haplogroup E. It was ancestral for Z1116 E1B1A1, CTS8649 E1B1B1B1, and could be designated as E1B1. The reason why finding E1B1 among the Natufian remains is important is because the Natufians are the ancestors of Semites. According to the book titled "In Hot Pursuit of Language in Prehistory," it reads, This paper makes an additional inference since there is archaeological and physical anthropological reason to believe that the Natufians were related to modern Semitic-speaking peoples of the Levant. And according to the book titled Transition to Modernity by Cambridge University, it reads, "Given the modern distribution of Semitic languages over the area in question, which belong to the larger Afro-Asiatic family, it seems appropriate to suggest that these first farmers of the Levant and their Mesolithic predecessors, including the Natufian culture, were speaking a Proto-Afro-Asiatic language. It may be that Proto-Semitic continued to develop within the Levant and Syria area, since both Ebelite and Akkadian early Semitic languages are attested earlier, the former around 2400 BC. Abraham could have been similar to this Natufian, having E1B1. There's a great article that actually talks about this, titled Resurrecting the Ancient Israelites from the Valley of Dry Bones. And it reads, using DNA to study Jewish ancestry, paleogenomics changed all that. Thanks to advances in the field, it became possible to extract DNA from ancient people and identify their mitochondrial haplogroups and even autosomal DNA. This remarkable process allowed the unthinkable, a semi-reveal of the ancient Israelites from their dry bones and the recovery of their stories. The DNA extracted from these bones can tell us who these people were and what they looked like and what they ate and what diseases they carried. We can trace their migration routes to gather a deeper understanding of where we all came from. Yes, they can also tell us how similar those ancient Israelites were to modern-day people, and if modern-day Jews are not the lineal descendants of the ancient Israelites, we can find who is. To answer some of these questions, I developed the primeval DNA test, which allows comparing the DNA of modern-day people to the ancient DNA extracted from skeletons of various people, including the ancient Israelites. The ancient Israelites were obtained from 3 regions: Matzah, Tachit, at the territory of the tribe of Benjamin, Pekin, Naphtali, and Raphat Cave, Manasseh, near the valley of the Raphat River. Other people may find that they are close to Abraham, a Turkish man, E1B1, who led a group of Anatolians to what he must have felt was the Promised Land. Interestingly, the Y-chromosomal haplotypes of the ancient Israelites are typically E1B1 and T1 haplotypes, commonly found today in Africa with lower frequencies in the Middle East and Europe. So there are scientific models in which Abraham and the Israelites carried E1B1. On a Jewish-owned genetic Facebook page titled Ancient Israelite DNA, there was an interesting discussion between the author of the page and another person in the thread about Abraham and the Israelites having E1B1. Josiah asked the host, I looked at a chart of the haplogroup E on Wikipedia and saw that there are 3 branches of E1B1, E1B1A, E1B1B, the Anatolian Abraham, and E1B1C. The E1B1B are the ancient Israelites, and the DNA which we will be compared to. ? Thank you again for your time and work. The host of the page replies saying, the ancient Israelites certainly in the test have E1B1, E1B1B1B2, and a lot of T's. Note that the Judeans had almost solely T. It's highly likely that Abraham and the Israelites would have carried E1B1. And this is interesting because if this is the case, then this means that the ancient Israelites would have been a mixture of both E1B1B and E1B1A populations, because E-P2 or E1B1 can produce both E1B1B and E1B1A. E-P2, also known as E1B1, is a human Y-chromosome DNA haplogroup. This paternal lineage has an ancient presence in the Middle East. Descendants E-V38 and E-M E-215, according to the paper titled A New Topology of the Human Y-Chromosome Haplogroup E1b1 E-P2, revealed through the use of newly characterized binary polymorphisms, and it reads, Haplogroup E1b1 now contains two basal branches, E-V38 E1b1a and E-M215 E1b1b, with E-V38/V100 joining the two previously separated lineages E-M2, formerly E1b1a, and E-M329, formerly E1b1c. And here is a chart from the paper showing how E1b1a and E1b1b both descend from P2 or E-P2, which is E1b1. Both of these lineages are the same. They have the same paternal forefather. With that being said, if Abraham was E-P2 or E1b1, he would produce both EV38 E1b1a and E-M215 E1b1b. This isn't hard to believe because we have people to this day born with E-P2 or E1b1. As an example, the Natufian remained in Israel. He had E-P2 or E1b1. Therefore, Abraham could have been similar to that Natufian with E-P2. Therefore, he would produce both E1b1a and E1b1b descendants. So Israel will be a mixture of E1b1a and E1b1b. As a matter of fact, geneticist Razib even stated in the past that the Israelites and ancient Semitic Middle Eastern populations could have had E1b1a among their populations. We can even find ancient E-M2 or E1b1a among the Arabian populations that's been there for over 2,000 years. If you go to SNP Tracker online right now, you can find Bronze Age and Iron Age and even Roman Age era E-M2 or E1b1a in the Arabian Peninsula and the Middle East. But here are two clips that will illustrate the ancient presence of haplogroup E, even E1b1a, in the Middle East.
There are groups that make claims for E1b1a being a founding haplogroup among Semitic groups. Is E1b1a found in high frequencies among Semitic populations like J1? Would the ancient Arabians and Syrians and Israelites have had high percentages of E1b1a within their populations in antiquity?
Yeah, they did, but they also had J1. So this is my opinion. So take that for what it is. I think that J1 and J2 probably were not, and I'm going to say Afro-Asiatic because, you know, like the Egyptians spoke non-Semitic language, but it was Afro-Asiatic, like more closely related to that of the Amazigh people, the Berbers, right? So North Africa had a different branch before the Semitic, whatever. So I'll say Afro-Asiatic, Semitic, and whatever the other groups like Tuareg and stuff. So it looks like the Natufians had it. It looks like there was a massive admixture between Western and Eastern Levant, Middle Eastern people. Probably before 3000 BC, but after, let's say after like 8000 BC, somewhere like in prehistory, there was a genetic exchange, and the J's, J1, J2, seem to show up during that exchange in the Levant and the western part of the Middle East. I don't think Afro-Asiatic languages are from Western Iran, which is where they would come from. Therefore, I don't think. Also, like you find J2 and J's in Europe and in India. And obviously, and that's not due to Semitic migration, right? So they're ancient lineages that are in like the Indus Valley civilization and what. Excuse me. So I think E is probably more likely to be Afro-Asiatic, and they had high frequencies in Arabians, Syrians, and Israelites.
Okay.
There's also the E haplogroup. There are Arabs who belong to the EM2 haplogroup specifically. That is the haplogroup identified with the Bantu expansion of sub-Saharan Africa. So originally, when some Arabs discovered that they were from EM2, but they weren't black looking, they were confused. They thought, oh, maybe my ancestor was originally enslaved from Africa and that's where my haplogroup comes from. But some of these people started working online, combining their ancestries, mainly on familytreedna.com, and discovered that these guys shared ancestry with other people at least 2,000 years old that was in the Arabian Peninsula. So 2,000 years ago, the E haplogroup somehow has been in the Arabian Peninsula. So the E haplogroup has been in the Arabian Peninsula for a very long time. In fact, there began to be a theory that the E haplogroup arrived in the Arabian Peninsula even before the J haplogroup returned, I'll talk about this later, from around Albania. But anyway, the point is, there's an ancient E haplogroup among the Arabs. And these people share absolutely no genetic similarities except for that haplogroup with Africans. So for example, if they go on 23andMe, it'll say 0.0% African DNA, but they'll belong to E. So this is a very ancient haplogroup among the Arabs.
So there is an ancient presence of haplogroup E, specifically E-M2, and haplogroup E1B1A could have been among the Israelites, and obviously, of course, E1B1B would be among ancient Arabs and the Israelites as well. With that being said, Abraham and the Israelites could have been E1B1. Therefore, the Israelites would be a mixture of E1B1A and E1B1B. The next possible candidate that Abraham and the Israelites could be is haplogroup E-M35/E-M215. One method we can use to determine the haplogroup of Abraham is by looking at subclades of the ancestors of Semitic populations, like the Natufians, and look at people who claim descent from the 12 tribes of Israel. One subclade of haplogroup E that Abraham could have been is a downstream of E-M35. E-M35 was found among the Natufian remains. The reason why finding E-M35 among the Natufian remains is important is because the Natufians are the ancestors of Semites. According to In Hot Pursuit of Language in Prehistory, it reads, "This paper makes an additional inference since there is archaeological and physical anthropological reason to believe that the Natufians were related to modern Semitic-speaking peoples of the Levant." And according to Cambridge University, it reads, "Given the modern distribution of Semitic languages over the area in question, which belong to the larger Afro-Asiatic family, it seems appropriate to suggest that these first farmers of the Levant and their Mesolithic predecessors, including the Natufian culture, were speaking a Proto-Afro-Asiatic language. It may be that Proto-Semitic continued to develop within the Levant and Syria area, since both Ebelite and Akkadian early Semitic languages are there attested earlier, the former around 2400 BC. And according to Y-chromosome diversity characterizes the Gulf of Oman, it reads, "The role of the Levant in the Neolithic dispersal of the E3B1-M35 sublineages is supported by the data." So haplogroup E-M35 has had a long and ancient presence in the Levant, which happens to also be the homeland of Semitic populations, where Semites will also disperse from the Levant going into Mesopotamia and Arabia. According to the Bayesian phylogenetic analysis of Semitic languages, identifies an early Bronze Age origin of Semitic in the Near East, and it reads, "We estimate that Semitic had an early Bronze Age origin approximately 5,750 years before presence in the Levant, followed by an expansion of Akkadian into Mesopotamia." With that being said, Abraham could have been EM35, which is also found among many Jewish populations and Arab populations today. E-M215 and E-M35 are quite common among Afro-Asiatic speakers. The linguistic group and carriers of E-M35 lineage have a high probability to have arisen and dispersed together from the Afro-Asiatic homeland. Amongst populations with an Afro-Asiatic speaking history, a significant portion of Jewish male lineages are E-M35. Haplogroup E-M35, which accounts for approximately 18 to 20% of Ashkenazi and 8.6% to 30% of Sephardi Y chromosomes. appears to be one of the major founding lineages of the Jewish population. According to Y-DNA research studies of rabbinical lineages and their importance to Jewish genealogy, it reads, "The following data tables, the figures summarize the data presented for each of the 13 rabbinical lineages in the previous section. Table 6 provides a summary of the defining haplogroup and subclade that is associated with the Y-DNA genetic signature for each of the 13 rabbinical lineages and the number of descendants tested and found to match the Y-DNA genetic signature." So down below, as you can see, Table 6, defining haplogroup and subclade for 13 rabbinical lineages. And as you can see, 3 of them have E-M35. Figure 1 presents a graphical depiction of the defining haplogroup and subclade for each of the 13 rabbinical lineages and the percentage of the total number of Y-DNA tested descendants in all studies belonging to each haplogroup. It does not reflect the percentages at which these haplogroups naturally occur in the Jewish population. Another 3 lineages, or nearly 1/4 of them, 23.1%, belong to the E-M35/E-L117 haplogroup. E-L117 is the previous haplogroup classification for E-M35. This is right in the middle of the range of 15 through 30% that has been cited as the percentage of all Jewish men who belong to the E1B1B E-M35 haplogroup. Table 7 presents a summary of the main number of genetic matches at 37 STR markers for the tested descendants for each of the 13 rabbinical lineages. These main numbers of genetic matches for each rabbinical lineage are depicted geographically in Figure 2. And as you can see down below in Table 7, the 3 families with E-M35 and the number of their descendants. As shown by the data in Table 7 and Figure 2, there is a great deal of variation in the number of genetic matches among the various haplogroups, ranging from a mean of 7 genetic matches for the lineage descending from Rabbi Yechiel Michael of Zlotchov E-M35 haplogroup to a main of 436 genetic matches for the Wertham Gitterman Hasidic dynasty E-L117 haplogroup. We might be able to even narrow down the specific subclade of E-M35 that Abraham could have been a part of by looking at Jewish populations with EM35. According to the paper titled Connections Between the Jewish Clusters and Subclades of Haplogroup E1B1B1, and it reads, "The existence of common SNP mutations for a number of clusters, subclades, and unclassified members of haplogroup E1B1B1 was suggested. TMRCA of common branches of several Jewish clusters was calculated. The hypothesis of their origin was put forward. There are 7 Jewish clusters in haplogroup E1B1B1, E1B1B1-C, E1B1B1-D, E1B1B1-D1, E1B1B1-A, E1B1B1-B, E1B1B1-C, and E1B1B1-E. These clusters include a total of about 20% of all Jewish men. It was of interest to determine their ancient origin, whether they are on the same branch with other unclassified haplotypes and clusters of haplogroup E1B1B1, mainly scattered in the Arabian Peninsula, North and Northeast Africa, and Western Europe, or with subclades whose geographical distribution is relatively localized, E1B1B1B, North Africa, and the maritime countries of Western Europe, and E1B1B1C, Eastern Mediterranean. The common ancestor of cluster E1b1b1-A and E1b1b1-B and a number of unclassified haplotypes of E1b1b1 lived 2,840 plus minus 920 years ago. That is, in the time of the existence of the Jewish nation. The common ancestor of this branch lived in the Middle East, judging from the geography of modern carriers of the haplogroup E1b1b1c1a. The presence of two Jewish clusters in this branch may be indicative of its Jewish origin. The common ancestor of cluster E1b1b1c1-D1 and the main branch of cluster E1b1b1c1-D2 lived 5,000 plus or minus 1,500 years ago. That is before the emergence of the Jewish nation. Judging from the geography of modern clusters of haplogroup E1b1b1c1, the common ancestor of this branch also lived in the Middle East, and its origin could be a Canaanite. The common ancestor of cluster E1b1b1-C and E1b1b1-D and two haplotypes E1b1b1 from Western Europe lived 5,000 plus or minus 1,600 years ago. The presence of this branch of two Jewish clusters and non-Jewish from Western Europe, as well as the origin of the haplotypes of the parent branch in the Arabian Peninsula, North Africa, and Western Europe, makes it impossible to draw a conclusion about the place of its origin. According to the paper titled TMRCA of Large Jewish Clusters of Haplogroup E1b1b1, and it reads, TMRCA of Jewish clusters E1b1b1 C1a-B and E1b1b1 C1-D1 has been updated based on the structure of their trees. A hypothesis on their origin in Europe has been put forward. It has been shown that these two clusters could not arisen simultaneously. There are 7 Jewish clusters in haplogroup E1b1b1, 2 large E1b1b1 C1-D1 and E1b1b1 C1a-B. Included a total of over 60% of the Jews of haplogroup E1b1b1, or 14% of all Jewish men. Members of these clusters share their places of origin Eastern and less frequently Western Europe. The samples represent the countries of origin of their members: Belarus, Poland, Lithuania, Russia, Ukraine, Germany, Great Britain, Italy, Holland, Austria-Hungary, Slovakia, Moldova, Armenia, and Israel. Single participants from Armenia and Israel might point to the migrations of the 20th century, not the era of clusters arising. TMRCA of cluster E1b1b1 depends on the tree shape and is within 1,280 through 1,840 years without confidence interval. That is, supposes the emergence of a cluster or in the Roman Empire, where most of the Jews were dispersed after the destruction of the Jerusalem Temple 70 CE, and especially after the defeat of Bar Kokhba uprising 135 CE, or in the early Middle Ages during the formation of the first Ashkenazi Jewish communities. So with all that being said, for the most part, one of the major haplogroups among the Jewish population is haplogroup E-M35. Most genetic papers admit this common fact, even when other haplogroups are mentioned, such as haplogroup J, T, or R. Haplogroup E is always seen as one of the major and main haplogroups out of the lesser ones. According to Genetic Ancestry Jewish from Jewish Virtual Library, it reads, haplogroup E-M35 and haplogroup J-12F2A fit the criteria for major Ashkenazi Jewish founding haplogroups because they are widespread both in Ashkenazi Jewish communities and in the Near East populations, and occurs at much lower frequencies in European non-Jewish populations. And according to the paper titled A Mosaic of People, the Jewish Story, and a Reassessment of the DNA Evidence, and it reads, an explanation of recent DNA studies clarifies the probable origins and history of haplogroup E3b among Jewish populations. One important study by Cruciani explores and refines the origins and distribution patterns not only of E3b, but of the entire E haplogroup. In a study that presented frequencies of haplogroups J and E among various groups, including both Ashkenazi and Sephardic populations, researchers found 14 out of 77 Ashkenazim, 18.2%, were E3b, while 12 out of 40 Sephardim were E3b, 30%. Bihar 2004 deemed subclade E-M35 a major founding lineage among Ashkenazim. And according to the paper titled Contrasting Patterns of Y-chromosome Variation in Ashkenazi Jewish and Host Non-Jewish European Populations, it reads, Para-group E-M35 and haplogroup J-12F2A fit the criteria for major Ashkenazi Jewish founding lineages because they are widespread both in Ashkenazi Jewish and in Near Eastern populations, and occur at much lower frequencies in European non-Jewish populations because they have similar distributions as these major founder lineages. And according to the paper titled Y-DNA Genetic Structure of the Sevran Bindi Hasidic Dynasty, and it reads, Genetic genealogy study focused on the Y-DNA pedigree of the Sevran Bindi Hasidic dynasty from Ukraine and Bessarabia during the 19th century. Genealogical and Y-chromosome genetic data are presented. All patrilineal descendants of the Wertham Gitterman rabbinical lineage share the L117 SNP of Y-DNA haplogroup E, previously described as E-M35 in the literature. Essential to determining the unique Y-DNA signature of a particular rabbinical lineage is the terminal single nucleotide polymorphism, SNP, which defines the haplogroup to which descendants of the lineage belong. In addition to having a high percentage of standard Y-DNA markers or short tandem repeats, STR, allele values in common. Another Y-DNA characteristic that all descendants of the Wertham Gitterman rabbinical lineage share is that they all belong to the E-L117 haplogroup. The E-L117 haplogroup was previously known as E-M35 and has been referred to in the literature as E1B1B1. According to the International Society of Genetic Genealogy, ISOGG, the National Geographic Genographic Project E1B1B1 may have arisen instead in the Near East or the Middle East and then expanded into the Mediterranean with the spread of agriculture. Haplogroup E-M35, which accounts for approximately 18 to 20% of Ashkenazi and 8.6 to 30% of Sephardi Y-chromosomes, appears to be one of the major founding lineages of the Jewish population. E-M35 has been observed in moderate numbers among individuals from Ashkenazi, Sephardic, and Samaritan communities having traditions of descending from the tribe of Levi, suggesting that the E1B1B1 men claiming to be Levites may have existed in Israel before the diaspora of 70 CE. If you go to Family Tree DNA and type in Jewish E Project Y-DNA Classic Chart, you will see that Ashkenazi, Sephardi, Mizrahi Jewish men around the world have haplogroup E. I took a few screenshots of the results, but if you go to the website, you'll get to see the full list of many Jewish people who in fact have haplogroup E. Jewish E Project formerly Jewish E3B project, Y-DNA Classic Chart. For genealogy within the recent 15 generations, STR markers help define paternal lineages. Y-DNA STR markers change, mutate often, enough that most men who share the same STR results also share a recent maternal lineage. This page displays Y-chromosome DNA, Y-DNA STR results for the project. It uses the classic format. The columns display each project member's kit number, paternal ancestry information, according to project settings, the paternal tree branch, haplogroup, and actual STR marker results. What's also fascinating about E-M35 is that it's not only seen among the Jewish population, but it's also seen among the Arabian population as well. If you type in yfull Y-tree-M35, you will find a map that illustrates a very high distribution of E-M35 in the Arabian Peninsula. Haplogroup E among Arab populations such as in Egypt can reach as high as 68.6%. Arabs in the Jordan of the Dead Sea can reach as high as 44.5% of haplogroup E. Then Arabs, Palestinian Christians can reach up to 31.8% of haplogroup E, and the list goes on. As a matter of fact, according to the paper titled The Genomic History of the Middle East, you can find many United Arab Emirates, Yemeni, Saudi Arabian, Iraqi, Iranian, Bedouin, Syrian, Jewish, and many other Middle Eastern populations that carry haplogroup E, and these E clades have been in the Middle East for thousands of years, according to their TMRCAs. As a matter of fact, if you type in Tufian's DNA, Y-DNA Classic Chart, you will find a list of various Middle Eastern populations with haplogroup E. If you go to SNP Tracker and type in E-M35, you'll see a very interesting distribution of haplogroup E in the Middle East and the Arabian Peninsula, as well as the Mediterranean, and E-M35 seems to have dispersed in the Levant, going all throughout the Fertile Crescent. With that being said, Abraham and the Israelites could have been a downstream of E-M35. Abraham and the Israelites could be a downstream of E-M78. One method we can use to determine the haplogroup of Abraham, and therefore the Israelites, is by looking at people today who claim descent from the 12 tribes of Israel. However, not just claim descent, but also have family records going all the way back to key figures in the Torah. The Samaritans in Israel from the family of Kohen, the tribe of Levi, trace their lineage back to the Levites. These Kohen Samaritans never left the land of Israel, and they have E-M78. Demographic investigations of the Samaritan community were carried out in the 1960s. Detailed pedigrees of the 13 generations show that the Samaritans comprise 4 lineages, one of them being our main focus, the priestly Kohen lineage from the tribe of Levi. Recently, several genetic studies on the Samaritan population were made using haplogroup comparisons as well as wide genome genetic studies. The biggest and most important Samaritan family, the Kohen family, tradition, tribe of Levi, was found to belong to haplogroup E. This article predated the change of the classification of haplogroup E3B1-M78 to E3B1A-M78, and the further subdivision of E3B1A-M78 into 6 subclades based on the research of Cruciani et al. According to Britannica, Samaritan, member of a community now nearly extinct that claims to be related by blood to those Israelites of ancient Samaria, who were not deported by the Assyrian conquerors of the kingdom of Israel in 722 BCE. According to the genetic paper titled Reconstruction of Patrilineages and Matrilineages of Samaritans and Other Israeli Populations from Y-chromosome and Mitochondrial DNA Sequences Variation, it reads, Haplogroup E3B1, defined by M78, was found in all 9 populations at frequencies of 10 through 16.7%. The Samaritan family Cohen carries this haplotype. A study was conducted on the Samaritans, and there are 4 Samaritan families who all claim different origins. However, it's the Cohen Samaritan family. This specific family is the one that claims to be from the tribe of Levi, and that Samaritan family has haplogroup E, specifically E-M78. According to the genetic paper titled Genetics and the History of the Samaritans, Y-chromosomal microsatellites and genetic affinities between Samaritans and Kohanim, and it reads, the Samaritans are a group of some 750 indigenous Middle Eastern people, about half of whom live in Holon, a suburb of Tel Aviv, and the other half live near Nablus. The results support the position of the Samaritans that they are descendants of the tribes of Israel dating to before the Assyrian exile in 722 through 720 BCE, in correspondence with previous published single nucleotide polymorphism haplotypes. Each Samaritan family, with the exception of the Samaritan Cohen lineage, was observed to carrying a distinctive Y-chromosome short tandem repeat haplotype that was not more than 1 mutation removed from the 6-marker Cohen model haplotype. In the Table 1 chart, it shows Cohen 1 and Cohen 2, and they have E3B, specifically E-M78. Cohen 1 and Cohen 2 are E-M78. And remember, Cohen, tribe of Levi. Furthermore, if you look on Family Tree DNA titled Jewish E-Project Y-DNA chart, you will see at the top the first sample is Israelite Samaritan tribe of Levi, and its haplogroup is haplogroup E, specifically E-FT161981. That specific subclade of E-M78 that the Samaritans have is found in the Jordan and Israel/Palestine, specifically Amman, Jordan, and specifically Palestine of Nablus. So E-FT161981 is definitely a Levantine haplogroup in the land of Israel and Jordan, exclusively restricted to this region and not seen anywhere else. Since the Samaritans carry E-M78 and claim the tribe of Levi, you could make the argument that if it goes back to that lineage, going back to Aaron, to Amram, to Kohath, to Levi, and then from Levi to Jacob, to Isaac, to Abraham. Abraham would represent a downstream of E-M78. Charts have been made to illustrate how this would work. Shem would be haplogroup DE, and then from Shem to Eber would be haplogroup E-P2 or E1B1, which can produce both E1B1A and E1B1B. Then Peleg or Ru would be E-M35, founding Jewish lineage. Then Ru or Serg would be E-M78, Abraham's lineage. Then all the way down to Abraham in 2000 BCE, he would be a downstream of E-M78. By the way, I didn't make this chart, but it is an interesting representation as to how Abraham could be E-M78. With that being said, Abraham and the Israelites would be E-M78, a downstream of it. basing this off of the Samaritan Kohan lineage of the tribe of Levi, and the Samaritan population has 100% haplogroup E. However, not just them, but also their Semitic cousins such as Arabs in Egypt, Arabs in the Jordan, Arabs in Palestine, etc. Haplogroup E-M78 is widespread throughout Africa, the Middle East, and the Mediterranean, and this lineage makes up a decent percentage in Israel today. With that being said, Abraham and the Israelites could represent downstream of E-M78. Abraham and the Israelites could be a downstream of E-V22. If Abraham is E-V22, then it's likely that he could descend from Akkadians and early Semitic populations that migrated into Mesopotamia from the Levant. According to the paper titled How Eurasia Was Born, it reads, "The most plausible candidates for Semitic remigration to the Fertile Crescent, with TMRCAs, the most common recent ancestors, fitting the arrival of the Akkadians and other early Semitic peoples, are certain subclades of both EV22 and EV12 with relatively early TMRCAs present in the Middle East could be candidates for such remigration, such as E-FGC14382, TMRCA is 2200 BCE, and E-V3262, TMRCA is 2600 BCE. Therefore, Abraham and his Semitic ancestors could have been EV22, who would have left the Levant and went into Mesopotamia. According to the genetic paper titled Bayesian Phylogenetic Analysis of Semitic Languages identifies an early Bronze Age origin of Semitic in the Near East, and it reads, "We estimate that Semitic had an early Bronze Age origin, approximately 5,750 years before present, in the Levant, followed by an expansion of Akkadian into Mesopotamia." So with all that being said, Abraham would descend from an EV22 lineage that left the Levant and migrated into Mesopotamia, like other Proto-Semites and downstreams of haplogroup E. This would mean that the Israelites would paternally be EV22. Modern Jewish populations do in fact carry EV22. According to the webpage Eupedia titled Haplogroup E1b1b, it reads, E-V22 is found primarily in western Ethiopia, northern Egypt, and in the southern Levant. In Europe, it is therefore associated with the Phoenicians and the Jews. The Phoenicians could have disseminated EV22 to Sicily, Sardinia, southern Spain, and the Maghreb, and the Jews too, to Greece and mainland Italy and Spain. According to the website titled JewishDNA.net, Jewish group of branches E1b-V22, it reads, "The Jewish branches can be grouped according to their Y-DNA characteristics. These characteristics are often related to a period between 10,000 YBP through 4,000 YBP. Some branches had a founding father effect in the period outside the Middle East, which gives information on the location of their ancestor at the time of the founding father. For other branches, it is more likely that the ancestors were in the Middle East, where in most cases no founding father effect took place and population growth was continuous. An overview of the Jewish groups of branches is present. Two likely origins for the Jewish V22 groups exist from Egypt or from Mesopotamia. Both areas are known to have V22 populations and large Jewish communities. And here is a chart showing the Jewish branches within EV22. So now let's take a look at some of these branches and read the information dealing with them. On your own time, you can click all of the links and read about each EV22. This branch has only one close branch according to Penelia. It consists of a few British and Dutch persons at about 1,700 years before present. A next branch is more than 3,300 years before present, away with people from a large region, Ethiopia, Saudi Arabia, Southern Europe, and Western Europe. In 2022, several people have done NGS, next generation sequencing. The old EV22 has a history as described on EV22.net. It originates in southern Egypt. The exchange of people between Egypt and the Levant is old, older than the start of Judaism. This branch has a large Ashkenazi and a smaller Sephardic branch. Whether the first Jewish people in this branch became Jewish in the Levant or in Egypt cannot yet be determined. This branch is in the E1B-V22 group of Jewish branches. This branch is a member of the Ashkenazi branches. This branch is a member of the Sephardic branches. This branch has some matches that might be related to the start of Judaism. If you look on Y heat map and type in E-V22, you will see that it's distributed throughout the Mediterranean as well as in the Middle East, and it is seen within the Arabian Peninsula, Mesopotamia, modern-day Iraq, as well as a very nice and high distribution in the Levant, or the land of Israel. What's also interesting about EV22 is that it's found among the Samaritan Cohen family from the tribe of Levi. According to Jewish Y-DNA branch, the owner of this kit reports himself as Cohen, tribe of Levi, Israelite Samaritan. This branch is in the E1B-V22 group of Jewish branches. This branch is a member of the Samaritan branches. Others are 149532 from an Israelite Samaritan tribe of Levi, Itmir, born Aaron Cohen. This branch is in the E1B-V22 group of Jewish branches. This branch is a member of the Sephardic branches. So as I stated before, EV22 is found heavily within Mesopotamia or modern-day Iraq, as well as in modern-day Israel, specifically among Jewish populations. EV22 is actually a downstream of E-M78. Therefore, Abraham, Isaac, and Jacob and Levi could be a downstream of EV22. The priestly Cohen lineage from the tribe of Levi. The biggest and most important Samaritan family, the Cohen family, tradition, tribe of Levi, was found to belong to Haplogroup E. This article predated the change to the classification of Haplogroup E3b1-M78 to E3b1a-M78, and the further subdivision of E3b1a-M78 into 6 subclades based on research of Cruciani et al. According to Britannica, Samaritan member, a community now nearly extinct that claims to be related by blood to those Israelites of ancient Samaria who were not deported by the Assyrian conquerors of the Kingdom of Israel in 722 BCE. And according to reconstruction of patrilineages and matrilineages of Samaritans and other Israeli populations from Y-chromosome and mitochondrial DNA sequence variation, it reads, Haplogroup E3b1, defined by M78, was found in all 9 populations at frequencies of 10 through 16.7%. The Samaritan family, Cohen, carries this haplotype. And if you go to Jewish E Project, formerly Jewish E3b Project, Y-DNA classic chart, you'll see Israelite Samaritan tribe of Levi, Aaron, Israel, as haplogroup E-FT161981. But with all that being said, Jews aren't the only Middle Eastern population with E-V22. Many Arabs also carry E-V22, which makes a lot of sense because of Ishmael. If you type in Y-full Y-tree E-V22, you'll see a very high distribution of EV22 in the Arabian Peninsula among Arab populations. Also, if you type in SNP Tracker, you'll see an even distribution of EV22 in both the Arabian Peninsula and the Middle East, as well as the Mediterranean. And the dispersion point for EV22 and all its descendants is again within the Levant, the homeland of Semitic populations. So plenty of Arab and Jewish populations carry haplogroup E, and Abraham could be a downstream of haplogroup EV22. The Israelites would be from this lineage. Therefore, here's one way you can look at things. Shem would be haplogroup DE. Then from Shem to Eber will be haplogroup E-P2, which can produce both E1B1A and E1B1B. Then Peleg or Ru would be haplogroup E-M35, and then from Peleg and Ru to Serg and Nahor, they will either produce EM78, and then from EM78 we'll have EV22, and Abraham will be a downstream of EV22. Therefore, with all of that being said, Abraham and the Israelites could have been a downstream of EV22. Abraham and the Israelites could be a downstream of E-V12. If Abraham is E-V12, then it's likely that he could be an Akkadian. If not Akkadian, then perhaps descendants of other early Semitic people in general. According to the paper How Eurasia Was Born, it reads, the most plausible candidates for Semitic remigration to the Fertile Crescent, with TMRCAs, the most common recent ancestors, fitting the arrival of the Akkadians and other early Semitic peoples, are certain subclades of both E-V22 and E-V12, with relatively early TMRCAs present in the Middle East, could be candidates for such remigration, such as E-FGC14382, TMRCAs 2200 BCE, and E-V3262, TMRCAs 2600 BCE. According to the Bayesian phylogenetic analysis of Semitic languages identifies an early Bronze Age origin of Semitic in the Near East, it reads, we estimate that Semitic had an early Bronze Age origin approximately 5,750 years before present in the Levant, followed by an expansion of Akkadian into Mesopotamia. So Abraham would descend from an E-V12 lineage that left the Levant and migrated into Mesopotamia, like other Proto-Semites, and would have been a downstream of haplogroup E. This would mean that the Israelites would paternally be E-V12, and modern Jewish populations do in fact carry E-V12. According to JewishDNA.net, Jewish group of branches E1B-V12, it reads, the Jewish branches can be grouped according to their Y DNA characteristics. These characteristics are often related to a period between 10,000 years before present through 4,000 years before present. Some branches had a founding father effect in that period outside the Middle East, which gives information to the location of their ancestor at the time of the founding father. For other branches, it is more likely that the ancestors were in the Middle East, where, in most cases, no founding father effect took place and population growth was continuous. An overview of the Jewish groups of branches is present. V12 has two major subgroups, V32 in the south or Arabia and CTS 693 in the area of Mesopotamia. All branches below are from the CTS 693 area, Mesopotamia. It seems likely that this is the area they became Jewish. So here are the branches in this group, the ones in Mesopotamia, the ones that are Jewish. E-V12 is distributed throughout the Middle East. There are various Mizrahi who are Middle Eastern Jews with EV12, but EV12 is also found in Ashkenazi and Sephardic populations as well, with their common ancestor living before the start of Judaism, 3,300 to 3,700 years before present. This would be around the time of the Bronze Age when Abraham lived. Could this common ancestor be Abraham? This branch is in the E1B-V12 group of Jewish branches. This branch is a member of the Mizrahi branches. The STR dataset of this branch is limited. No NGS test is known. It is possible that this branch is a shared ancestor with AB-141 after the start of Judaism. This branch is in the E1B-V12 group of Jewish branches. This branch is a member of the Mizrahi branches. In the Sephardic page of FTDNA, we find 122368 and 89104, both from Iraq, but less than 67 markers, close to E1B-V12, YB8540. The shared ancestor SNP is at least CTS6667, which is 3,300 years before present on yfull.com. Likely that the shared ancestor is more recent and they share a Jewish ancestor. This branch is in the E1B-V12 group of Jewish branches. This branch is a member of the Ashkenazi branches. This branch is a member of the Sephardic branches. This branch has some matches that might be related after the start of Judaism. This branch from Sephardic origin has several Jewish persons in Iraq and Turkey. In 2019, we have Spanish and Sephardic descendants in this branch with different surnames in both groups and STR distances that indicate a TMRCA, the most common recent ancestor, of more than 500 years and less than 1,000 years. This branch has a shared ancestor in YFull CTS8411 and FTDNA with two other branches. This SNP is about 3,700 years before present and has another Jewish branch and an Irish branch. It is most likely that CTS8411 was in the Levant, and 3 descendant lines are measured. One left to the Ashkenazi countries, one went to Spain being Jewish, and one ended up in Ireland. In Spain, a few descendants left Iberia as Sephardic Jews, and a few others became Catholic and remained in Iberia. The next closest member in YFull are 2 persons from Sardinia. Given the history of Sardinia and the Levant, one could speculate that they arrived in Sardinia in the Phoenician period. This branch is in the E1B-V12 group of Jewish branches. This branch is a member of the Sephardic branches. Interestingly enough, we see that the Laura rabbinical family has one of the oldest family trees in the world, tracing their lineage back to King David, has EV12. 2 Ashkenazi E1B-V12 groups came possibly from Iberia. The Greek person has a surname, Ashkenazi, and is probably from Ashkenazi origin. The FTDNA Laura project has several descendants from the line Laura, as reported on encyclopedia.com. The information on this site and other sites gives Samson Laura, born near the Windsensee 1325 and became rabbi in Erfurt. This branch is in the E1B-V12 group of Jewish branches. This branch is a member of the Ashkenazi branches. According to the paper titled Why DNA Research Studies of Rabbinical Lineages and Their Importance to Jewish Genealogy, it reads, the Laura rabbinical family has one of the oldest family trees in the world, claiming to trace back at least to King David, born circa 1037 BCE. The first Laura in the lineage was Rabbi Aaron Laura, circa 1400 through 1480, who married Miriam Shapira, the daughter of Rabbi Shlomo Shapira, the founder of the Shapira rabbinical lineage. The predicted haplogroup to which the 2 pedigreed Laura descendants belong is E-M35. The previous nomenclature for this haplogroup, E1B1B1, was cited for Laura's descendants, the Rabbi Jekyll Laura, circa 1435 through 1490. There's a great book on this family titled The Laura Legacy, the House of Davidic Royal Descent, and it reads, the author of The Unbroken Chain has just written another major work, this time about one of the most distinguished Jewish families, Laura. It is a very ancient family, tracing its roots back through Rashi and Hillel to King David. The family is known throughout history for its extraordinary achievements in science, philosophy, theology, and the arts. The book is a comprehensive study of the Laura family that includes 45 pages of family trees showing the relationship of the Laura family to such other families. Even if you go to the DavidicDynasty.com, where they trace and keep the records of people who have descent from King David, it reads, "The Laura Legacy, the House of Davidic Royal Descent" by Nell Rosenstein compiles the sources of Rashi and hence Davidic descent of major rabbinic families through the Laura family. The Laura family is a very ancient family, tracing its roots back to Rashi and Hillel to King David. If you go to Family Tree DNA titled The Laura DNA Project Y-DNA Classic Chart, you will find that most are haplogroup E with a minor J and R. The Laura DNA Project Y-DNA Classic Chart. For genealogy within the most recent 15 generations, STR markers help define paternal lineages. Y-DNA STR markers change, mutate, often enough that most men who share the same STR results also share a recent paternal lineage. This page displays Y-chromosome DNA, Y-DNA STR results for the project. It uses the classic format. The columns display each project member's kit number, paternal ancestry information according to project settings, the paternal tree branch haplogroup, and actual STR marker results. So here are a few screenshots of the Laura rabbinical family's DNA, and when it comes to their E markers, and you can just look it up yourself and see all the various E markers under the Laura rabbinical family. This is fascinating because if the Laura rabbinical family traces their lineage back to King David, King David's lineage traces back to Judah, then Judah to Jacob, Isaac, and Abraham, meaning Abraham is E-V12 and paternally Israelites are E-V12. Abraham would represent a specific downstream of E-V12. Therefore, the way this could work is Shem is haplogroup DE, then Eber is haplogroup E-P2, which can produce both E1B1A and E1B1B. Peleg or Ru would be E-M35, then Ru or Serg would be E-M78, then Serg or Nahor would be E-V12, and then all the way from Serg to Abraham, he would be a downstream of E-V12. With that being said, Abraham and the Israelites could be E-V12. One candidate for the haplogroup of Abraham and the Israelites is E-Z830. One method we can use to determine the haplogroup of Abraham is by looking at subclades of the ancestors of Semitic populations like the Natufians, and look at people who claim descent from the 12 tribes of Israel. One subclade of haplogroup E that Abraham could have is a downstream of E-Z830. E-Z830 was found among the Natufian remains. According to the supplementary information in the paper titled The Genetic Structure of the World's First Farmers, it shows 2 E1B1B2 samples. According to the genetic analysis done on 6 Natufian remains from northern Israel, the Natufians carried the Y-DNA haplogroup E-Z830, a somewhat upwind clade of E-M123, and therefore ancestral to it. The Natufians were one of the first settled peoples in the world and may have contributed to the domestication of certain crops and thus the advent of agriculture. The discovery of E-Z830 without other clades suggests an indigenous presence in Canaan and Israel that predates all other clades, which are not known to have existed in the region at that time, 10,000 years before present. E-M123 is thought to have a TMRCA, the most common recent ancestor, about 18,000 years ago, 8,000 years before the Natufians, possibly ancestral remains are from. Haplogroup E1B1B1B2 has been found in 75% of the N-Gazelle population, along with 60% of PPNB populations, circa 8,800 through 6,500 BC. So haplogroup E was abundant among the Natufians, but specifically E-Z830 was abundant among the pre-pottery Neolithic populations as well as the Natufian populations. The reason why finding E-Z830 among the Natufians is important is because the Natufians are the ancestors of Semites. According to the book In Hot Pursuit of Language in Prehistory, it reads, "This paper makes an additional inference since there is archaeological and physical anthropological reason to believe that the Natufians were related to modern Semitic-speaking peoples of the Levant." And according to Cambridge University, it reads, "Given the modern distribution of Semitic languages over the area in question, which belong to the larger Afro-Asiatic family, it seems appropriate to suggest that these first farmers of the Levant and their Mesolithic predecessors, including the Natufian culture, were speaking a Proto-Afro-Asiatic language. It may be that Proto-Semitic continued to develop within the Levant and Syria area, since both Ebolites and Akkadian, early Semitic languages, are there attested earlier, the former around 2400 BC. If Abraham descended from Natufians with E-Z830, then Abraham and the Israelites would be a downstream of E-Z830. Many Jewish and Arab populations carry E-Z830. A recently confirmed subclade of E-Z827, Z830, includes the confirmed subclades of E-M123, E-M293, and E-V42, and is a sibling clade to E-L19. Currently, the E-M35 phylogeny project recognizes 4 distinct clusters of Z830 carriers, 2 of which are exclusively Jewish in origin. The remaining 2 are significantly smaller and include scattered individuals in Germany, Spain, Latin America, Egypt, and Ethiopia. Cluster name E-PF1975, ISOGG nomenclature E1B1B1B2. Description: this cluster is now characterized by SNP PF1975. Formerly, this was known as cluster E-Z830-A Jewish cluster. Cluster name E-PF1952, ISOGG nomenclature E1B1B1B2. Description: this cluster is now characterized by SNP PF1952. Formerly, this is known as cluster E-Z830-B Jewish cluster. Cluster name E-Z830-C ISOGG nomenclature E1B1B1B2. Name cluster E-Z830 ISOGG nomenclature E1B1B1B2. What's interesting about this subclade of haplogroup E is that famous Jews such as Albert Einstein carried it. According to iGena, titled The DNA Profile of Albert Einstein, it reads, "Why DNA analysis with various members of Albert Einstein's paternal lineage were able to determine his haplogroup. The result also confirmed his Jewish ancestry. Albert Einstein belonged to haplogroup E, subgroup E-PF1952 in his paternal lineage. The acclaimed theoretical physicist Albert Einstein is presumed to have belonged to Y haplogroup E-Z830, based on the results from a patrilineal descendant of Naphtali Hasher Einstein. Albert Einstein's great-grandfather Approximately 20% of Ashkenazi Jews belong to E1b1b. Here are a few interesting Jewish clusters within E-Z830 according to JewishDNA.net. This branch has two ancestors, AB-008 and AB-009. This distance between the two is not yet determined. Most likely, the shared ancestor of the two lived in the Ashkenazi or Sephardic countries. This branch has three lines, England, Italy, Switzerland, and is estimated by Henlea at 2,100 years before present. It is likely this line went from the Levant to Iberia to the two Ashkenazi branches. This branch is in the E1b-old group of Jewish branches. This branch is a member of the Ashkenazi branches. This branch has some matches that might be related after the start of Judaism. Looking at one more interesting subclade within E-Z830 that's part of the Jewish cluster from JewishDNA.net, it reads, "This group is a Ashkenazi group that has no close connection. The faraway connection are more than 3,000 years before present, possibly from the Middle East, where other Z830 groups are probably originating. This branch is in the E1b old group of Jewish branches. This branch is a member of the Ashkenazi branches. E-Z830 is also quite common among Arabian populations as well. If you type in E-Z830 on yful.com, you will see a very high percentage of this subclade within the Arabian Peninsula. Also, if you type in Y heat map E-Z830, you'll still see a very high percentage within the Arabian Peninsula and throughout the Mediterranean and other parts of the Middle East, even in Africa. And the same thing occurs if you type in E-Z830 into SNP Tracker. You'll see very high distribution within the Arabian Peninsula and other parts of the Middle East, as well as the Mediterranean and even Europe. And the split seems to occur within the Levant, with branches going south into the Middle East and Arabia and branches going north into Europe. With all of that being said, Abraham and the Israelites could be a downstream of E-Z830. One candidate that Abraham and the Israelites could be is E-M123. One method we can use to determine the haplogroup of Abraham and therefore the Israelites is by looking at the origin of the subclade and the location for the origin of Semites. One subclade of haplogroup E that Abraham could have been is a downstream of E-M123. E-M123 is a downstream of E-Z830, which the Natufians carried. According to the genetic analysis done on 6 Natufian remains from northern Israel, the Natufians carried the Y DNA haplogroup E-Z830, a somewhat upwind clade of E-M123, and therefore ancestral to it. The Natufians were one of the first settled peoples in the world, and may have contributed to the domestication of certain crops and thus the advent of agriculture. The discovery of E-Z830 without other clades suggests an indigenous presence in Canaan and Israel that predates all other clades, which are not known to have existed in the region at that time, 10,000 years before present. E-M123 is thought to have a TMRCA, the most common recent ancestor, about 18,000 years ago. 8,000 years before the Natufians, possibly ancestral remains are from. Haplogroup E1b1b12 have been found in 75% of the N-Gazelle population, along with 60% of PP&B populations, circa 8,800 through 6,500 BC. According to the book In Hot Pursuit of Language and Prehistory, it reads, "This paper makes an additional inference since there is archaeological and physical anthropological reason to believe that the Natufians were related to modern Semitic-speaking peoples of the Levant. And according to Cambridge University, it reads, given the modern distribution of Semitic languages over the area in question, which belong to the larger Afro-Asiatic family, it seems appropriate to suggest that these first farmers of the Levant and their Mesolithic predecessors, including the Natufian culture, were speaking a Proto-Afro-Asiatic language. It may be that Proto-Semitic continued to develop within the Levant and Syria area, since both Ebulites and Akkadian, early Semitic languages, are there attested early, the former around 2400 BC. So we find the ancestral haplogroup of E-M123, which is E-Z830, among the Natufian remains. Furthermore, E-M123 has a Levantine origin, which is important considering that the Levant is the origin and homeland of Proto-Semites. According to the webpage Eupedia titled E1B1b, it reads, E-M123 originated some 19,000 years ago during the last ice age. Its place of origin is uncertain, but it was probably in the Red Sea region, somewhere between the southern Levant and Ethiopia. Its main subclade E-M34 most probably emerged in the Levant about 15,000 years ago. Soon afterwards, M34 split into two branches, M84 and Z841, which were probably found in the Fertile Crescent during the Neolithic period. And according to the webpage This Week in Palestine, titled Rooted in the Holy Land, it reads, when tests of the deep clade group examine the more recent mutations of your paternal ancestors, the E-M123 clade indicates the Levant and Anatolia as the most recent region of origin, and we have found samples of E-M123 and members of that branch within the Levant as well as in Anatolia around the time of the early Bronze Age and late Chalcolithic. And finding haplogroup E in the Levant is important because Semites will expand from the Levant into Mesopotamia. According to the paper titled Bayesian Phylogenetic Analysis of Semitic Languages identifies an early Bronze Age origin of Semitic in the Near East, and it reads, we estimate that Semitic had an early Bronze Age origin approximately 5,750 years before present in the Levant, followed by an expansion of Akkadian into Mesopotamia. So as we can see, Semites will expand from the Levant going into Mesopotamia and Arabia, where we find high amounts of haplogroup E. Therefore, we can come to the conclusion that these Proto-Semitic or early Semitic populations would have been carrying E markers as they expanded from the Levant, where there was high amounts of haplogroup E. When it comes to modern populations and ancient populations, E-M123 is found among many Jewish and Arab populations and is a great candidate for an Israelite haplogroup. According to the paper titled A Mosaic of People, the Jewish Story and a Reassessment of the DNA Evidence, and it reads, in fact, the best candidates for possibly E3b Israelite ancestry among Jews is E-M123. This subclade occurs in almost the same proportions, approximately 10 through 12%, among both Ashkenazim and Sephardim. According to Christiani in 2004, E-M123 probably originated in the Middle East, since it is found in a large majority of the population from that area, and then back migrated to Ethiopia. Yet, the fact that both Sephardim and Ashkenazim possess this subclade in similar high frequency supports an Israelite/Middle Eastern origin. And according to the page titled Why DNA Haplogroup E, E1B1b, and E1B1a, it reads, this haplogroup has a long presence in the Near East as well, as demonstrated by its occurrence in ancient remains from the Levant and mummies in Egypt. Today, a significant contingent of Ashkenazi, 20%, and Sephardi, 30%, Jews belong to a specific sub-haplogroup of E-M215, defined by the mutation E-M123, supporting this as a major founding lineage of the Jewish people. And according to the paper titled Reconstruction of Patrilineages and Matrilineages of Samaritans and Other Israeli Populations from Y-chromosome and Mitochondrial DNA Sequences Variation, it reads, Haplogroup E3B3, defined by M123, was present at 5 to 20% in Ethiopian, Ashkenazi, Libyan, and Yemenite Jews and in Palestinians. Arabian populations also carry E-M123 as well, in parts of Yemen especially, as high as 25 to 50%. E-M123 is best known for its major subclade E-M34, which dominates this clade. However, earlier studies did not test for E-M34. Looking beyond its geographical patterns, E-M123 is also quite common in many Semitic-speaking communities, including among Ashkenazi and Sephardic Jews, accounting for over 10% to all male lines. But more importantly, looking at the chart and the map, we see that Yemenites can carry up to 25% to 50% of haplogroup E, specifically E-M123, within E-M34. If you look on yful.com and type in E-M123, you can find it among Mediterranean populations, but highest in the Arabian Peninsula among Arab populations, as well as throughout the Middle East and even in parts of Africa. If you type in Y heat map, you'll also see the exact same thing. Distributions of E-M123 throughout the Mediterranean, but highest in the Middle East, specifically the Arabian Peninsula and Israel and other parts of the Near East. Also, if you type in SNP Tracker E-M123, you'll see an even split of E-M123 within the Middle East, highest in the Arabian Peninsula, but also throughout the Mediterranean and Europe and parts of Africa. And the split seems to occur within the Levant, with groups going south into the Middle East and groups going north into the Mediterranean. With all of that being said, Abraham and the Israelites could be a downstream of E-M123. One candidate for Abraham and the Israelites is E-M34. One method we can use to determine the haplogroup of Abraham and therefore the Israelites is by looking at the origin of the subclade and the location for the origin of Semites. One subclade of haplogroup E that Abraham could have been is a downstream of E-M34. E-M34 is a downstream of E-M123 and E-Z830, which the Natufians carried. According to the webpage Eupedia titled E1B1B, it reads, E-M34 lineages experience a much more dramatic expansion during the Chalcolithic Copper Age period. Also, downstream of CTS1096 and Y14891 and Z21018 clades are typically found among people of Jewish ancestry, while PF6391 and Z21421 are found in the Levant, the Syria, Lebanon, Palestine, Jordan, and the Arabian Peninsula. F1382 appears to have expanded during the Iron Age from the Levant to the Arabian Peninsula, where it is almost exclusively found today. And in this time period of the early Bronze Age, we can find many E-M34 samples throughout the Middle East, such as in the Levant and Syria near Ebla, which was an ancient Semitic kingdom. They were an Eastern Semitic speaking people, but also in Anatolia. Could Abraham also be one of these Semitic populations with E-M34? This is actually a high probability. According to the paper titled Haplogroups M34 and M84 among Jews, could Abraham be E1B1B1C1 or E1B1B1C1A? The present paper clarifies the TMRCA, the most common recent ancestor of the Jews of haplogroup E1B1B1C1, the origin of Jews of haplogroup M84, and answer the question, could Abraham be M34 or M84? The specific structure of Jewish sample of haplogroup E1b1b1c1, the vast majority of the haplotypes belong to one cluster, makes the TMRCA very sensitive to including of any new haplotype not belonging to that cluster. Due to the instability of the TMRCA of the Jewish E1b1c1, the authors emphasize the importance of the confidence interval calculated by 95% probability. Also, the origin of Jews of haplogroup M84, which has the largest number of clusters, has not been adequately studied so far. In addition, the simultaneous presence of M34 subclades among the Jews and Arabs contribute to the assumption of belonging Y-chromosomal Abraham, a conditional common ancestor of the Arabs and Jews, and one of these subclades. The question of the lifetime of common ancestors of Jews and Arabs of haplogroups E1b1c1 and E1b1c1a has not been studied yet. The aim of this paper is to clarify the TMRCA of the Jews of haplogroup E1b1c1, the origin of Jews of haplogroup M84, and answer the question, could Abraham be M34 or M84? The most common ancestors of Jews of E1b1c1 and E1b1c1a. For our research, we will use Jewish E1b1c1 haplotypes from Haplozone E-M34 database. The calculation according to the algorithm shows that the recent common ancestor of the sample lived 5,650 plus or minus 2,820 years ago, with the probability of 95%. To determine the TMRCA of Jews of haplogroup M84, we use haplotypes of the Jewish clusters E1b1b1c1a-A, E1b1b1c1a-B, E1b1b1c1a-C from Haplozone E-M35 database. Our calculation shows that the TMRCA of the sample is 4,100 plus or minus 1,740 years with 95% probability. At the time of writing the paper, July 2010, in Sharif DNA Project and Cohen DNA Project, there were 3 haplotypes of E1b1b1c1a Saids and 2 haplotypes of E1b1b1c1 Cohens, as well as dozens of Arabian and Jewish E1b1c1a and E1b1c1 haplotypes. Haplotypes of E1b1c1 Saids and E1b1c1a Cohens are not yet available. The TMRCAs of Arabian and Jewish E1b1c1 and E1b1c1a are 8,080 plus or minus 3,890 and 4,080 plus or minus 1,440 years ago. Consequently, the Jewish and Arabian lines of E1b1c1 divided one from the other several thousands of years before biblical Abraham. On the other hand, the common ancestor of the Jewish and Arabian E1b1c1a lived in the same historical era as the biblical Abraham. The result demonstrates the close affinity of Jewish and Arabian E1b1c1a. The TMRCA of E1b1c1a Saids is 4,080 plus or minus 1,560 years ago. That more than thousands of years over the era of Ali and excludes their Said origin. Conclusions. 1. Subclades E1b1c1 and E1b1c1a were included in Jewish community during the conquest of Canaan. With 95% probability, the TMRCA of Jewish E1b1c1 and E1b1c1a are 5,650 plus or minus 2,820 and 4,100 plus or minus 1,740 years. 2. Subclades E1b1c1 and E1b1c1a both found among Jews and Arabs, including a small number of Cohens and Saids. The calculated TMRCAs show that the most recent E1b1c1 ancestor of Arabs and Jews lived 8,080 plus or minus 3,890 years ago. And the most recent E1B1C1A ancestor of Arabs and Jews lived 4,080 plus or minus 1,440 years ago. The most recent common ancestor of E1B1C1A, Said, lived 4,080 plus or minus 1,560 years ago. The most recent common ancestor of E1B1C1, Cohens, lived 440 plus or minus 410 years ago. 3. Team RCA calculations show that according to formal characteristics, subclades E1B1C1 cannot claim to be the Abraham's haplogroup. 4. The most recent common ancestor of the Jewish and Arabian E1B1C1A lived in the same historical era as the biblical Abraham, but a contradiction of the calculated Team RCA of Cohens and Saids to historical data exclude haplogroup E1B1C1A from the contenders of the role of Abraham's haplogroup. Despite this, the results indicate close relationship of Jewish and Arabian E1B1C1A lines. By the way, when it mentioned Saids having EM34 and EM84 that's related to the Jews, a Said is a honorific title denoting people accepted as descendants of the Islamic prophet Abraham through his grandson Hassan ibn Ali and Hassan ibn Ali, sons of Abraham's daughter Fatima and his cousin and son-in-law Ali. And according to Britannica, Said, Arabic title of respect sometimes restricted as is the title Sharf in the Banu Hashim, members of Muhammad's clan, in particular the descendants of Muhammad's uncle Al-Abbasa and Abu Talib and of Ali ibn Abi Talib. By Muhammad's daughter Fatima in the Hijaz, Said is further restricted to the descendants of Hassan, the younger son of Ali and Fatima. Also, Sharfs were mentioned as having EM34 and EM84 in the paper and being related to the Jews. A Sharf is a term used to designate a person descendant or claiming to be descendant from the family of the Islamic prophet Muhammad. In the broader sense, it refers to any descendant of Abraham's great-grandfather Hasim, the Banu Hasim or Hasimites, already in Muhammad's day an established clan within the Meccan tribe of the Quraysh, including all descendants of Muhammad's paternal uncles Abu Talib and Al-Abbasa. More often, it refers to a descendant of Ali, a son of Abu Talib, and a paternal cousin of Muhammad, especially not exclusively through Ali's marriage with Muhammad's daughter Fatima. In its narrowest sense, it refers only to someone who descends from Ali and Fatima's eldest son and Muhammad's grandson Hassan. And according to Britannica.com, Sharif, Arabic title of respect restricted after the advent of Islam to members of Muhammad's clan of Hasim, in particular to descendants of his uncle Abi Abbas and Abu Talib, and of the latter's son Ali by Muhammad's daughter Fatima. In the Hijaz, the title of Sharifs is said to have been further restricted to the descendants of Hassan, the elder son of Ali and Fatima. Sharifs originally were heads of prominent families in a town. Therefore, according to the family tree of the Safavids and the Saids, and they are related to the Jews, and both the Jews and these Arabs carry E-M 34 and 84. And of course, both Jews and Arabs have a tradition of descending from Abraham. And there just so happens to be a subclade of haplogroup E that both the Arabs and the Jews descend from that was in the Bronze Age when Abraham would have lived. Could this subclade of E be Abraham? But let's continue reading. According to the paper titled, "How Did So Much Middle Eastern DNA Get to Wales?" examining 3 possible sources: Carthaginians, Romans, English Jews. And it reads, E-M 34 is prevalent among Ashkenazi and Sephardic Jews, as well as among Ethiopians. That both Ashkenazic and Sephardic Jews carry this haplotype indicates that it entered the Levantine Jewish community early on, and then spread outward as Jews split into Sephardic and Ashkenazic lineages. According to Family Tree DNA titled E-Y6923 Jewish Haplogroup, it reads, "This project is for men who have done Y DNA testing, and their terminal haplogroup designation by Family Tree DNA will be either E-Y6923 or one of its downstream branches, E-L791, or it will be predicted E-M35 or E-M34, with Y matches who are E-Y6923 and /or E-L791. The Y6923 SNP is found downstream of E-L791 and likely occurred about 1,700 to 1,600 years ago. Its main Ashkenazi branch, defined by E-Y6940, dates to about 1,250 to 1,100 years ago. It seems to have been one of the major founding branches of early Ashkenazi community. Other branches which have been found in Libyan Jews, Algerian Jews, Tunisian Jews, Greek Jews, Sicilians, Puerto Ricans, New Mexicans, Peruvians, Moselle Germans, and French Canadians include E-Y102667, E-FT227227, E-FT405052, and one yet unnamed branch. Clearly, EM34 is among Jewish populations, but let's look deeper into Arabian populations that carry EM34. According to the paper titled Arabian Clusters of Haplogroup M34, it reads, Haplogroup M34 and its subclade M84 were detected among the Arabs in the Arabian Peninsula. A possible reason for migration of the founder of cluster E1B1B1C1A-E from the Levant to the Arabian Peninsula could be the Crusades. The highest diversity of subclades of haplogroup M34 is observed in the Levant and Anatolia. Therefore, its ancestral home is often placed in the eastern Mediterranean. In addition, haplogroup M34 and its subclade M84 were detected among the Arabs in the Arabian Peninsula, where they form specific clusters E1B1C1-B and E1B1C1A-E. Knowing the age of the clusters and their area of distribution, we can find the history of clusters' origin and resettlement of their carriers. In this paper, we will try to find out the history of E1B1C1 and E1B1C1A subclades in the Arabian Peninsula on the example of these clusters. The sample of cluster E1B1C1A-E consists of 567 marker haplotypes. This cluster TMRCA is 1090 plus minus 510 years. Despite the fact, due to different sizes of samples, the ages of these clusters' founders are different. It should examine the entire period of their confidence intervals, which are intersected. It is possible that both clusters have arisen in about the same medieval era and are linked to the migration of their founders from the Levant to the Arabian Peninsula, according to some important event. What could have caused this migration? We think that a possible cause of the medieval migrations from the Levant could be the Crusades, a series of Western invasions to oust the Muslims from Palestine, which lasted almost 200 years, 1096 through 1272 years. The First Crusade ended with the capture of Jerusalem and the massacre of Muslims. Apparently, these invasions, and as a result of them, looting and killing, forced part of the Muslims to seek refuge from persecution of the Crusaders closer to Mecca. This, in our view, could cause to arise at least one cluster of Arabia E1B1C1A-E. Conclusion: 1. Carriers of subclades M34 and M84 identified in the Arabian Peninsula, where they form clusters E1B1C1-B and E1B1C1A-E. 2. The TMRCA of cluster E1B1C1-B is 50 plus or minus 320 years ago. The TMRCA of cluster E1B1C1A-E is 1090 plus or minus 510 years ago. They possibly arose at one time. 3. A possible reason for migration of the founder of cluster E1B1C1A-E from the Levant to the Arabian Peninsula could be the Crusades. With all of that being said, we can find a very high distribution of EM34 in the Arabian Peninsula. It represents at around 25 to 50% in Yemenites as an example. If you take a look at Yful.com and type in E-M34, you can see a high percentage of this haplogroup within the Arabian Peninsula, as well as other parts of the Middle East and the Mediterranean. Also, if you type in Y heat map E-M34, you see a high distribution within the Arabian Peninsula, other parts of the Middle East, and the Mediterranean. Also, if you take a look at SNP Tracker and type in E-M34, you'll see a high distribution in the Arabian Peninsula as well as in other parts of the Middle East and the Mediterranean, and it seems that the split would have took place in the Levant, with groups of Semites going into the Middle East of Arabia and other groups of Semites staying in the Levant and others expanding to the Mediterranean and Europe. With that being said, I will say that E-M34 is a great candidate for Abraham and the Israelites. Abraham and Israel could be E-M84. One method we can use to determine the haplogroup of Abraham and therefore the Israelites is by looking at the origin of the subclade and the location for the origin of Semites. One subclade of haplogroup E that Abraham could have been is a downstream of E-M84. E-M84 is a downstream of E-M34 to E-M123 and to E-Z830, which the Natufians carried. Abraham could have been a downstream of E-M84. E-M84 has been found in the early Bronze Age of Israel, Syria and Ebla, Turkey and Anatolia, and even Armenia. From a study on population genomics of Anatolia, the Caucasus, and the Levant, found in the ancient city of Ebla during the Bronze Age, one male individual who is E-M84. A study on population genomics of Anatolia, the Caucasus, and the Levant found in Aristalep one individual who lived circa 3369 through 3110 BC who belonged to E-M84. A study on the population genomics of Bronze Age Eurasia found in the remains from Nurken-Kitashan in Armenia lived during the Middle Bronze Age to E-M84. So around the time period of the early Bronze Age in the Middle East, you have plenty of E-M84 samples likely belonging to Semitic populations and descending from ancient Natufian populations that were in the Levant. Could Abraham be one of these E-M84 or be similar to a person with E-M84 around this time period? When it comes to modern populations with E-M84 such as Jews and Arabs, many carry this haplogroup. According to JewishDNA.net, all of these groups are M84 and probably originated in the Middle East. This branch is in the E1B-M84 group of Jewish branches. Let's actually read some of these branches. This age is estimated at 3,200 years before present, a period where many branch splits occur in the Middle East and descendants to the Mediterranean. It has early branches to Italy, at least 3, Portugal, and the Ashkenazi branch. This branch is in the E1B-M84 group of Jewish branches. This branch is a member of the Ashkenazi branches. The genealogy of this person from the Babylonian community from Baghdad and can be found on this website. This branch is in the E1B-M84 group of Jewish branches. This branch is a member of the Mizrahi branches. This branch is in the E1B-M84 group of Jewish branches. This branch is a member of the Mizrahi branches. This branch is in the E1b-M84 group of Jewish branches. This branch is a member of the Sephardic branches. This branch is a member of the Romanite branches. This branch is in the E1b-M84 group of Jewish branches. This branch is a member of the Mountain Jews branches. This branch has some branches that might be related after the start of Judaism. Many Arabian populations also carry E-M84, such as the Yemenites, who can carry around 25 to 50%. There are various Arab clans who carry E-M84 all throughout the Arabian Peninsula. If you take a look at yful.com and type in E-M84, you'll see a very high distribution in the Arabian Peninsula, Yemen, Kuwait, but also throughout other parts of the Middle East, such as Iraq, Israel, Syria, and parts of the Mediterranean. Also, if you type in Y heat map E-M84, you'll see again high distribution of this haplogroup, of this subclade within Israel, the Arabian Peninsula, Kuwait, Oman, Yemen, and even parts of Eastern Africa, but also throughout the Mediterranean world as well. And if you type in on SNP Tracker E-M84, you'll see a split of Semites from the Levant going into the Arabian Peninsula, and from the Levant going into the Mediterranean and Europe, as well as other parts of the Middle East. With that being said, Abraham and the Israelites could be a downstream of E-M84. What is the best Y-DNA haplogroup for Abraham and the Israelites? My top 3 haplogroups for Abraham and the Israelites, starting with number 1, is E1b1, also known as E-P2, and then for number 2, out of my top 3 is E-M35, but specifically E-M78 and its subclades. For number 3 out of my top 3 is E-M35, specifically E-Z830 and its subclades. Starting with number 3, E-Z830 is the third best candidate for Abraham and the Israelites. The Israelites would represent some subclade within E-Z830. The reason I say this is because many Jews and Arabs are under E-Z830. E-Z830 is even considered to be one of the major founding lineages of the Jewish population. According to the paper titled A Mosaic of People, the Jewish Story and a Reassessment of the DNA Evidence, it reads, in fact, the best candidate for possibly E3b Israelite ancestry among the Jews is E-M123. This subclade occurs in almost the same proportions, approximately 10 through 12%, among both Ashkenazim and Sephardim. According to Cruciani in 2004, E-M123 probably originated in the Middle East, since it is found in a large majority of the population from that area, and then back migrated to Ethiopia. Yet, the fact that both Sephardim and Ashkenazim possess this subclade in similar high frequency supports an Israelite/Middle Eastern origin. And according to Y-DNA haplogroup E, E1b1b, and E1b1a, it reads, this haplogroup has a long presence in the Near East as well, as demonstrated by its occurrence in ancient remains in the Levant and mummies in Egypt. Today, a significant contingent of Ashkenazi 20% and Sephardi 30% Jews belong to a specific sub-haplogroup of E-M215, defined by the mutation E-M123, supporting this as a major founding lineage of the Jewish people. As you can see, E-Z830 is a great candidate for the Israelites and the Ishmaelites. If you type in SNP Tracker, you will see various subclades of E-Z830 in the Arabian Peninsula and other parts of the Middle East, but you'll also see a very high distribution of these subclades around the Mediterranean and in Europe, and the split seems to occur in the Levant, in the Middle East, where both the Israelites and the Ishmaelites and other descendants of Abraham would have been and would have migrated from. Therefore, I say E-Z830 is the third best candidate for Abraham's haplogroup and therefore the Israelites. E-M78 is the second best candidate for Abraham and the Israelites. The Israelites would represent some subclade of E-M78. The reason I say this is because the Samaritan Levites in Israel carry this haplogroup, as well as the lower rabbinical family who claim descent from King David and have one of the oldest family trees in the world, also descends from E-M78. According to the paper titled Why DNA Research Studies of Rabbinical Lineages and Their Importance to Jewish Genealogy, reads, the Laura rabbinical lineage has one of the oldest family trees in the world, claiming to trace back at least to King David, born circa 1037 BCE. The first Laura in the lineage was Rabbi Aaron Laura, circa 1400 through 1480, who married Miriam Pura, the daughter of Rabbi Shlomo Pura, the founder of the Shapiro rabbinical lineage. The predicted haplogroup to which the two pedigreed Laura descendants belong is E-M35. The previous nomenclature for this haplogroup, E1B1B1, was cited for Laura's descendants of Rabbi Jerichel Laura, circa 1435 through 1490. And according to JewishDNA.net, the FTDNA Laura project has several descendants from the line Laura, as reported on encyclopedia.com. The information on this and other sites gives Samson Laura, born near Westlacey 1325, and became rabbi in Erfurt. This branch is in the E1B-V12 group of Jewish branches. This branch is a member of the Ashkenazi branches. And according to Davidic dynasty, which records all the descendants of King David, it reads, the Laura legacy, the house of Davidic royal descent by Nell Rosenstein, compiles the sources for Rashi and hence Davidic descent of the major rabbinic families through the Laura family. The Laura family is a very ancient family, tracing its roots back through Rashi and Hillel to King David. Now for the Levites. The priestly Kohen lineage from the tribe of Levi, the biggest and most important Samaritan family, the Kohen family, tradition, tribe of Levi, was found to belong to haplogroup E. This article predated the change of the classification of haplogroup E3B1-M78 to E3B1A-M78, and the further subdivision of E3B1A-M78 into 6 subclades based on the research of Cruciani et al. And according to JewishDNA.net, it says, the owner of this kit reports himself as Kohen, tribe of Levi, Israelite Samaritan. This branch is in the E1B-V22 group of Jewish branches. This branch is a member of the Samaritan branches. Therefore, Abraham could be a downstream of E-M78, either EV22 or EV12. If you type in SNP Tracker E-M78, you see a distribution of E-M78 in the Middle East, specifically the Arabian Peninsula, the Arabs, but also in the Mediterranean and Europe with the Jews. And the split occurs in the Levant, which is where the Ishmaelites and Israelites were located. With that being said, E-M78 is the second best candidate I have for Abraham and therefore the Israelites. Now, with all of that being said, the number one best candidate I have for the haplogroup of Abraham is E1B1, also known as E-P2. If Abraham is E1B1, not only would Israel be downstreams of E1B1A and E1B1B, this also includes Ishmaelite Arabs, Midianites, Moabites, Ammonites, and Edomites, and other descendants of Abraham. All of these groups could represent some individual subclade under E1B1 or E-P2. Abraham as E1B1 leaves more possibilities open for who might be Israel. And it's not just me saying this. There are papers that illustrate that Abraham could be E1B1. According to the article titled Resurrecting the Ancient Israelites from the Valley of Dry Bones, it reads, other people may find that they are close to Abraham, a Turkish man, E1B1, who led a group of Anatolians to what he might have thought was the Promised Land. Interestingly, the Y chromosomal haplotype of the ancient Israelites are typically E1B1, and T1 haplotypes, commonly found today in Africa with lower frequencies in the Middle East and Europe. And on a Jewish-owned Facebook page titled Ancient Israelites DNA, the admin and another person were discussing the likely haplogroup of Abraham and the Israelites. Josiah says, I looked at a chart of the haplogroup E on Wikipedia and saw that there are 3 branches of E1b1, E1b1a, E1b1b, the Anatolian Abraham, and E1b1c. The E1b1b are the ancient Israelites and the DNA which we will be compared to, ? Then the admin of the page responds saying, the ancient Israelites currently in the test have E1b1, E1b1b2, and a lot of T's. Note that the Judeans had almost solely T. If Abraham truly is E1b1, this would mean that the Israelites are a mixture of E1b1a and E1b1b. If you take a look at SNP Tracker and type in E-P2, you see it spread throughout the Middle East, Africa, and Europe, but its origin point is in the Levant. Therefore, I say E-P2 is the best candidate for Abraham, since his descendants could represent various subclades within this branch, and therefore the Israelites could be a mixture of E1b1a and E1b1b. With that being said, have a blessed day and shalom.